Discover the AutoLys Workstation, one of the new generation of smart automation solutions from Hamilton Robotics. The AutoLys Workstation is the first fully automated and compact system to automate the complete sample lysis and DNA purification process, thus removing the last remaining bottleneck of the DNA purification process. The AutoLys Workstation guarantees sample integrity and traceability throughout the lysis process. The AutoLys Tube Smart Design allows complete workflow automation in a fully enclosed and compact workstation, thus eliminating all tedious and labor-intensive manual sample lysis steps. The AutoLys Tube is a smart tube-in-tube device with 2D barcode identification at the bottom of the outer tube and a non-DNA binding filter membrane inside the inner tube. Once the sample is placed in the AutoLys tube, manual handling is not required throughout the entire lysis process, thereby guaranteeing the complete automation and sample integrity. After the initial incubation with shaking required by the sample lysis, the AutoLys tube is transformed into a spin basket by special AutoLys channels without the human intervention. The resulting lysate is then filtered into the outer tube by centrifugation to guarantee the best sample recovery and highest yields. To facilitate the tube handling, a special AutoLys rack is used, allowing handling of 24 tubes at a time. These AutoLys racks are in SBS format, making them compatible with centrifuges and 2D barcode readers, as well as Hamilton automated storage systems. AutoLys tubes also feature a special blue cap to facilitate easy manual handling for a manual workflow. The AutoLys tubes are placed in the AutoLys racks after manually inserting the raw samples into the tubes and matching the evidence sample barcode and the AutoLys 2D barcode. When loading the AutoLys racks on the robot, the AutoLoad barcode reading ensures a high level of verification to prevent possible human error. The EasyCode 2D barcode reader will identify and verify the individual AutoLys tubes. AutoLys channels will open and close the inner tube to allow the addition of reagents like lysis buffer. These same channels also transport tubes individually to a heater shaker, specially designed to offer the best heat transfer performance during the lysis incubation time. After lysis incubation, the shaking stops and the AutoLys channels transport tubes back to their original position in the AutoLys racks on the robot deck. The AutoLys channels then perform the lift and lock function, transforming the AutoLys tubes into spin baskets by lifting the inner tube and locking it in position. To separate the lysate from cellular debris and sample support, for example swabs, cigarette butts, and any other solid material, the AutoLys racks are centrifuged. The lysate goes through the sterile filter membrane of the inner tube, resulting in a clear lysate in the outer tube. After centrifugation, the racks are transferred back to their original positions on the robot deck. To access the clear lysate, the inner tubes are lifted and removed. The pipetting channels then transfer the clear lysates from the outer tubes to the destination format of your choice, for example, tubes or plates. The sample lysis process now is completed. Optionally, you may continue with DNA purification in this same method run. The AutoLys Workstation offers the time-saving benefit of performing the sample lysis process completely unattended. Tedious manual handling steps are eliminated. It minimizes the risk of contamination from human error. There is total traceability, and chain of custody is maintained by barcode reading. Whatever your throughput needs, whether 24 or up to 96 samples or more with a single run, AutoLys Workstation can be adapted to meet your needs with a guarantee of sample integrity and high DNA yields.